Yeah, my talk's going to be of an area of science that my group's been involved in for about 10 years now. We've got into thinking about how peptides, biological peptides, could have come to be part of the biological system. There's lots of really interesting questions here, and these questions really closely feed into the concept of origins of life. And so from our perspective, we've heard various things about planet formation this morning, and I certainly know very, very little about this. But from our perspective, the context of this kind of plot of Earth's history is most important, how we think about it, in the fact that as we go back in time, over eons, through billions of years, the record of life's history gets less and less and less clear. Rocks are eroded, the context, the information we have about the chemical systems that could have led to life in these early points are less and less robust. They get to the point where we just don't know the chemical inputs. We don't have a set of chemical materials that we can say definitely went in to the reactions that gave life. So that is sad. We can't just rerun the origin experiment because we don't know what it was. We have another record of life's history, the phylogenetic record, and this again can be used, the information in modern genomes, the fact that we know this information passed from mother to daughter over the same kind of plot of history. We can use this to trace the history of life as well as the fossil record. But again, we have a problem in the context of getting back to the origins of life experiment through this data, and that this is, as we make this biological regression backwards towards the origins of life, we can simplify in one respect, but in a molecular respect, the point at which all of the current genomes of all of the existing organisms on the planet, we regress back to is this molecule here. This is a ribosome. It is life's most complicated molecular machine. That is one reason to put it up. It is also life's molecular machine for making peptides. Right? This is a very complicated kind of 250,000 atom structure that is the full regression of the data we have from a biological perspective. So it is chemistry, life's most complicated machine. It is as far back as we can get with the regression of comparative biology. So again, like with the fossil record, we have this problem that we have a great data set to interrogate life's history, but that doesn't get anywhere close to assessing the origin of life. So, two data sets, no data on the actual origins chemistry that could have led to the process that would set off evolution to life as we know it. So how do we address this problem? Well, in chemistry, we can address this problem in a different way. We can tackle this, and this is what we do in my lab, as an engineering problem. How would we rebuild that system from scratch? Right? So if we cannot discover the origin of life, as beautifully put by Eshimosa here, can we rebuild it? Can we redesign that system? Can we re-engineer the chemical inputs that are required to set us on a pathway to make life from simple components? And this is an interesting dynamic in this group, right? Because what we're specifically trying to do is decouple our reactions from those historical events. We're trying to ask, what does it take to rebuild this system de novo, irrespective of those historical analyses? Those historical analyses can't tell us about that origins event, so we have to set up as many potential pathways and evaluate them as possible to build new data sets to ask what this origins event could have been. So generally speaking, and I'm going to focus on one aspect of this, interested in all different aspects, we're looking to establish a network of chemical reactions, a network of chemical interactions that can display the phenotypic properties expected of biology. Right? So we're going to need a system that can bring in nutrient, energy, stuff, material, carbon, nitrogen, the things of life from the environment. It's going to have to use those to build components that can influence information and function. Right? We would call these genomes and enzymes, put whatever terms you want on them in biological context, but this is the system we want to re-establish. And we could go very broad scale here, use any chemical components we want, but what we're going to do is we're going to focus on the chemical solutions that biology used. Right? So information is stored in nucleic acids. These are very specific chemical structures that are universal to life on Earth. And catalysis is run by sometimes those nucleic acids, but by very specific peptide structures that are universal all the way back to that last common ancestor, to that process of the ribosomal peptide synthesis. So can we understand how simple chemical input could come in to build the molecules that we've put a little bit of molecular detail on here, nucleic acids and protein molecules, and ultimately couple their synthesis so that the information in this system can control the phenotype in this system? And can we build that from scratch? So there's lots of different aspects. And as I said, I'm going to tell you a little bit about the work we've done towards peptides, the protein elements of life. And so why we became fascinated with this area is fairly easy to explain. In origins of life studies, we suffer from an n=1 problem all the time. Right? One tree of life, one form of ribosome, one genetic code. But in biology, there are two processes, not both universal, but there are two processes to build peptide molecules. Okay, the one we've met is the ribosome again, ribosomal peptide synthesis that takes an mRNA code, uses tRNAs in this huge kind of catalytic organelle, the ribosome, to build specific structured functional peptide molecules. This one is obviously older than the second system. The second system is uninventively named non-ribosomal peptide synthesis because it doesn't use a ribosome, but it uses the products of the ribosome, so large protein constructs, to synthesize peptide molecules. And these obviously have different evolutionary histories. One is dependent on the other, but one did not evolve from the other. Absolutely clear that that is not true. So these chemical systems are really different, right? In one of these systems, the ribosome is a large construct of mostly nucleic acid molecule, right? That uses nucleic acids for coding, for decoding, and for catalysis. In the non-ribosomal form, these are peptide molecules, right? That are using peptides for the same type of processes. In this system, the chemistries are different. So we have ester activation of peptide, we have thioester activation of peptide. But what is absolutely the same is the fact that both of these syntheses operate by the same strategy. They move from the N-terminus of a peptide to the C-terminus. They activate the growing polymer to addition of an incoming nucleophilic monomer. And that is the same across both of these systems. So we're no longer in an n=1 system. Why does biology, why did biology choose this strategy for peptide synthesis not once, but twice? What are the advantages of that? Why would we become obsessed by this problem as chemists? Because it is absolutely not what chemists do. So chemical peptide synthesis works in the exact opposite way. You work from the C-terminus to the N-terminus, you activate the monomer to addition of a nucleophilic polymer. It's the exact opposite of biology. So it's a really interesting question. It's a question that can tell us something about the evolution, the origin, the chemical underpinnings of life. Why does life choose this strategy twice, and why do chemists choose this strategy time and time again? So we think about what it takes to make a peptide in the context of rebuilding a cell de novo. So in a very simple sense, we could start with amino acids. Our problem here is we're going uphill, right? Chemically, we're trying to push this enormous rock up this hill to make peptide. Another way of thinking about this in the context of the system we're trying to make, what we're trying to do here is feed waste in the bottom of this process, right? In the middle of here, we have active functional biopolymers, right? They will hydrolyze over time for some of these eons, for some of these more quickly, to waste products. The waste product of our biopolymer peptides are the amino acids, right? So another way of thinking about this is what do we have to feed in the context of nutrient to be able to make these as a metastable state en route to amino acid waste? Where do we get that energy from? Well, we first considered, there are other ways to consider this, my lab actually considered other ways, what stable energy-rich molecules can an environment potentially feed? Purely abiotic molecules. So they definitely don't come from biology, but they could potentially be used to build biology. And we've had discussions of this context. These are the kind of molecules that can be found and maybe, or maybe not, delivered to the early Earth from space. We also, very, very briefly, have already considered these same kinds of activations in the context of nucleic acid synthesis, right? So in the same way, if we're trying to build a nucleic acid, not doing it from hydrolyzed sugar molecules, you can use nitriles to overcome this problem, the energy in nitriles, to build nucleic acids step by step without coming through these kind of hydrolyzed waste end products of this reaction. Okay, so we have a molecule we want to build, peptide molecules. We have a potential mechanism to have high energy molecules, nitriles, that can feed this process. Is there anything else that sits in our favor? Well, the oldest organic chemistry reaction in organic chemistry literature, first developed in the 1820s, is the Strecker reaction, right? A simple aldehyde or ketone, aldehydes give proteinogenic amino acids, and cyanide and ammonia will react to form an aminonitrile. This aminonitrile then hydrolyzes to form an amino acid. So this is the same output as hydrolysis of peptide would be. We have a pathway, simple, chemically robust, that will give intermediates that are activated with respect to that amino acid. Could these be the molecules that feed the synthesis, are the nutrient for peptide synthesis at the outset of biochemistry? They've got the energy, they've got another advantage, okay? So to make a peptide bond, our amine, our incoming nucleophile, which was our monomer in biological peptide synthesis, must be nucleophilic. To be nucleophilic, it must have electrons on this nitrogen that are available to donate to something else. It has these if it's a free amine. It doesn't have these if it's a protonated ammonium. Okay? So in biologically relevant pHs, the amino acid substrate has a high pK near 10. So across the biologically relevant pHs, it is protonated, its nucleophilicity is quenched. The aminonitrile, because of this nitrile group that brings energy that we could use for peptide synthesis, is strongly electron withdrawing. So maybe counterintuitively, by pulling electrons away from this amine, we make it more nucleophilic, more available to act, to deliver electrons from this nitrogen, because its pK drops from near 10 to near 5. So across the full pH range that we would be thinking about in the context of a cell, in the context of biology, we're going to have active nucleophile. So, background, actual chemistry. If we take an aminonitrile, we can put that in water with an activated acid. We're going to use a thioacid. I'll show you why in 2 slides' time. This will couple quantitatively in water. The aminonitrile as an amine is significantly, much more nucleophilic than the water. So we take an activated acid, we activate this generally by oxidation, there's lots of ways of doing this, and it will couple quantitatively to make an amide bond. When we make that amide bond, we switch this electron demand, right? So this nitrile's electron withdrawing, the amine is pushing electrons this way. As we form an amide, electrons start to flow the other way. This activates this nitrile that's been added to this amide as an electrophile. So then we can add hydrogen sulfide, forms a thioamide, which will hydrolyze to another thioacid. This thioacid is then at the C-terminus, the growing terminus of our peptide chain, and you can then iterate that cycle. Put in more aminonitrile, oxidize your thioacid, and you can then build a ligation cycle that is directed to follow the biological pathway from N to C terminus, where the energy to build the peptide bond is delivered by this stable abiotic nitrile motif. This process works remarkably well across the board for all amino acids. Very boring table of very high yields. But let's look at just one of those, right? So if you look at lysine, which is arguably the most challenging amino acid to couple into a native peptide, because it has two amines. It's got the alpha amine that has to be coupled into our peptide backbone, and it has an epsilon amine, the red one here, that shouldn't be coupled, that should stay as a free amine to allow us to have lysine side chains in our peptides, in our proteinogenic peptides. If you try to run these reactions at any pH you want, it is incredibly difficult, very, very difficult to differentiate these two amines. They're essentially equally basic, so they're equally protonated. So if you try to couple the lysine amino acid, where X is an acid, or a lysine peptide, where this is an amide, you get very little differentiation. You get a mixture of proteinogenic, blue coupling, and non-proteinogenic side chain derivatization that we arguably don't want to be able to make lysine with these structures. This differentiation is really difficult because these two amines are essentially the same basicity, the same nucleophilicity. If you move to the aminonitrile, the pK difference is enormous. It's like nearly 6 units difference, a logarithmic scale. At neutral pH, this amine is free. At neutral pH, this amine is near completely protonated. So at neutral pH, you only couple through this alpha amine, the biological coupling mechanism, directed again by that nitrile, by that activation of that amine through the electron withdrawing activating group that is added, that is bringing the energy for peptide synthesis. You can take those coupled lysines, you can thylize, hydrolyze, and then couple again through to make peptide bonds with that lysine side chain intact. If you try and shorten this side chain though, so biological proteinogenic lysine, 5-carbon backbone or side chain, ornithine is also biological. This is an important precursor of proline. This is in cells. But if you try this N to C terminal coupling through ornithine, it is totally different, night and day. Because of this shorter side chain, with exactly the same pK, so equal protonation of these two residues, the ornithine will quantitatively cyclize, blocking that N to C terminal ligation, whereas lysine will get through, will quantitatively allow peptide synthesis. So a purely chemical mechanism to differentiate between biological but non-proteinogenic ornithine and biological proteinogenic lysine. So something really interesting that jumped out about us in this chemical synthesis in general, as well as it spontaneously leading to that N to C terminal direction, as well as being able to deliver the chemical activation for peptide synthesis in one step, is that this residue is kinetically stable. Right? So when we add it, our first addition product, we activate our peptide residue to couple our aminonitrile, we get a stable product. This is why all our yields are enormously high. We can wait until the reaction finishes. This product will do nothing. It will form the peptide bond we need, and then it will sit there and it will wait until we change the conditions. Useful synthetically, but also this is really important in the context of biochemistry, in the context of what catalysis and phenotype is going to do. So in our system, I haven't got the system to flick back to quickly here, there were interconnections between these molecules. These are nodes of control where catalysis has to affect control between different molecules. And what's really important in this system, and it's just like ATP in biology, is this incoming monomer is thermodynamically activated but kinetically stable. This means it is amenable to catalytic control. Okay, so with the right catalyst, which is for this system as simple as substituting hydrogen sulfide for a sulfide catalyst, so a thiol residue, one can then make this type of coupling with the nitrile residue catalytic and catalytically controlled. So you can take the peptide nitrile products of our coupling strategy with an amine nucleophile. They will do nothing on their own. But if you add a thiol catalyst at neutral pH, the thiol will intercept the nitrile, and then this activated intermediate will couple with the amine to switch on this catalysis. This catalysis requires that this nitrile is somewhat activated by that electron withdrawal process I showed you earlier on, where electrons are flowing away from this carbon here. So this catalytic peptide coupling with a simple thiol catalyst will operate for alpha, proteinogenic amino acid nitriles, but not for beta. Right? This non-proteinogenic, but still natural, still biological residue will not couple through this activation strategy because this nitrile is not sufficiently activated to induce the catalyst to add and then allow the peptide ligation process to happen. When this ligation happens, if the ligation partner is a natural proteinogenic peptide, then this peptide will induce catalytic hydrolysis of our intermediate to deliver selectively peptide through organo, quote unquote, catalysis in water at neutral pH. Without that natural peptide framework, so if we couple a longer peptide, for example, here, without that catalysis, that intramolecular catalysis, hydrolysis is dominant, not peptide formation. So again, another strategy that is directed by the intrinsic nature of the peptide molecules, those that will become proteinogenic, to deliver peptides over their counterpart, non-proteinogenic versions. And again, this boring table of results. So again, we have a system, we're building a system, we're trying to think about this in the context of how to rebuild life. We have a catalyst concept, a simple thiol, that could run a catalytic peptide ligation. We can ask, which substrate would biology use to catalyze this kind of reaction? And there's absolutely one winner that you would pick in the context of all biology to catalyze activated acyl transfer, and that is this molecule. It is obviously a thiol. It's going to fit with our catalytic strategy. This is CoA, but the bit we're really interested in is this bit in bold, which is called pantethine. This is biology's universal acyl transfer cofactor. Every organism uses it. It is just as universal as genetics, as our alpha proteinogenic peptides, but importantly for us, it's outside our initial analysis. Okay, so we had a gene-centric analysis. We're going to look at where proteinogenic peptides came from because we're going to couple them to genes. This is integral to life's metabolism, but isn't part of that genetic process. So we can bring this in. We can ask questions. This allows us to ask new questions about our prebiotic model. And the question that is obvious to ask is, if our nitriles are predisposed to give proteinogenic amino acids, are they also predisposed to deliver pantethine? Now, these are totally different molecules, right? This has beta peptides that I've shown you don't couple through catalytic ligation, has alpha hydroxyls and quirky relationships that aren't expressed up here. So why should chemistry specifically deliver these molecules and also specifically deliver this molecule? Seems improbable at the outset, but obviously I'm going to show you it works, otherwise I wouldn't be bringing it up. So if we take our same precursors, the same aldehydes that can give proteinogenic amino acids, and put them through the Strecker reaction, obviously they only give Strecker products. It's beautifully clean. But if we take those aldehydes and pre-incubate them, pH 7, phosphate buffer, we see a new product arise. So two of our precursor aldehydes, the precursor of glycine and valine, undergo selective reaction together to form a new aldehyde. That aldehyde also undergoes cyanohydrin formation to make a cyanohydrin that is the nitrile activated form of pantoic acid. That's the first residue of pantethine. Right? This spontaneously forms from these proteinogenic amino acid precursors. If we then add the ammonia that will build the aminonitriles from our proteinogenic precursors, the pantethine remains differentiated. So while ammonia can work its way into the alpha amine positions for the proteinogenic amino acids, because of this additional hydroxyl that was added in our reaction in the first step, we cannot add an amine at this position. This hydroxyl cyclizes onto that nitrile before the oxygen can exchange, which means because we added this group, because it became pantethine, it cannot form alpha amines anymore. It cannot become an alpha amino acid. It gets stuck here, but it only gets stuck here temporarily. If we add a beta aminonitrile, and remember, just to remind you, you can form this perfectly adequately in the presence of alpha aminonitriles. If you add a beta aminonitrile, then you get equilibrium exchange with this amidine structure to then give the amine addition, hydrolysis, and the nitrile activated form of pantothenic acid, the second intermediate on pantethine synthesis. This is highly selective for the beta amine over the alpha amine. If the alpha amine does go in, it gets totally capped through a cyclization process. So if any alpha amine, proteinogenic amine, makes it into this pathway, it gets blocked. It can't go any further through this synthesis pathway. Obviously, our substrate is a beta nitrile, so it will not couple to alpha proteinogenic amino acid type nucleophiles in our catalytic peptide ligation strategies. What we need to get coupling intramolecularize this reaction. So if our catalytic thiol is attached through carbon to the amine nucleophile, then we can go through a 5-membered intramolecular intermediate that allows the coupling of this beta nitrile to a amine nucleophile. This amine nucleophile then has to have a beta thiol, basically has to be cysteamine. So you have to deliver pantethine as a product of this reaction. At neutral pH, this reaction then becomes quantitative and selective. Okay, so we think then that pantethine is a product of the same chemistry, the same nitrile chemistry that could spontaneously lead to proteinogenic peptides. Where do we go from here? Well, where I want to take you to here is back to our original problem. How do we address the origins of proteinogenic peptides when what we have is the ribosome as the end of our biological information? And there's something that I've totally ignored, right? In the context of how and why biology makes peptides, it is making them to express phenotypic function from genes. To do this, what we need to do is couple an amino acid to a nucleic acid so that nucleic acids can coordinate the synthesis of proteinogenic amino acids. So what we have here is, again, another representation of ribosomal peptide synthesis, now separated out into 2 steps. We have a step where we have a tRNA. This is going to be our adapter molecule. This allows us to translate genetic information into proteinogenic information. An enzyme is going to load an amino acid specifically to the 3' terminus of this RNA. that then allows this RNA to direct within the ribosome peptide synthesis. But hopefully you can see here our problem. Classic origins of life problem, we've got a chicken and egg problem. We have protein molecules that are directing the synthesis of protein molecules. I need ribosomal peptide synthesis to make these, but I need these to make ribosomal peptide synthesis. How do we break out of this chicken and egg problem? Well, this molecule does it. So biology's universal acyl transfer cofactor, this thiol, will react and does. This is the intermediate of non-ribosomal peptide synthesis. It will form activated thioesters with amino acids and will deliver this activated amino acid selectively to the diol of an RNA. So biological activation of amino acid directs nucleic acids to intercept activated amino acids en route to becoming polypeptides. These are really stable, neutral pH. They're really selective at neutral pH, so they don't react well with amines themselves, they don't react well with phosphates, they don't react well with imidazoles. What they react with really well with are low pKa alcohols, such as the diol of RNA. And the diol of RNA specifically over its 5' hydroxyl, for example, or the diol of RNA over the monool of DNA. Right? So we have a biological substrate that will direct the aminoacylation of biology's amino acid carrier that will lead it then into coded protein synthesis. The amount that we get is essentially just dependent on the amount of thioester we have. More thioester, more ester. There are lots of ways to induce this chemistry. But for example, you can take really dilute nucleic acid, really dilute aminothioester, freeze these into a eutectic solution and drive really effective, high-yielding aminoacylation of nucleotides from very low concentrations of both of our substrates. In the context of specific aminoacylation of RNA, we have a problem. The problem is that RNA has a ton of hydroxyls. Right? It's got one terminal diol that we should load amino acid to, but it has hydroxyls at every residue. And the longer an RNA gets, the more our selectivity challenge appears. So if we take single-strand RNA, thioester activated amino acid, we essentially aminoacylate it all over the place. The way to direct the amino acid to the correct position, to the C-terminus, is very simple. It's very biological. It's Watson-Crick base pairing. As the duplex forms, this inhibits the reaction at the internal residues, but it doesn't impact our reaction at the terminal residue. So double-strand RNA will chemoselectively react, aminoacylate at the 3' terminus, as would be required for a tRNA. And this duplex formation has no inhibitory effect on that terminal position. Doesn't matter whether it's an overhang, a nick, in any position, as long as the diol is free, there is limited effect to what duplex does. And this chemistry works across the board. You know, amino acids tested are all roughly comparable in the context of their aminoacylation, with the exception of arginine. So arginine is special here. It is roughly an order of magnitude more reactive than the others because it cyclizes intramolecularly, allowing this intermediate to undergo more extensive, more rapid aminoacylation. It's a steady state reaction. The faster the on rate, the higher our yield, but it doesn't induce any more peptide synthesis or remains selective for aminoacylation. This substrate uses our thiol. It can again become catalytic. So our thiol can be a catalytic acyl transfer agent if we take a generically activated amino acid, here an NCA, put it into water, it will generically hydrolyze, quantitatively. This is what is going to happen. If we add the thiol will trap that amino acid faster than hydrolysis. The thiol is really nucleophilic. If we swap our thiol for a nucleotide, we see no aminoacylation. It's a terrible reaction. Basically, our amino acid hydrolyzes. Couple these two processes together. If we have activated amino acid and acyl transfer cofactor, so pantothene, in the presence of a nucleic acid, the thiol will intercept the activated amino acid, generate in situ a biologically activated amino acid, which will transfer that amino acid to the terminus of RNA. You can use biological activation, you can use chemical activation, and you can get aminonitriles, albeit not in situ. I can talk to you about this offline if you want, to form the same structures where essentially all pathways lead to that same kind of process, directing aminoacylation. So the thing I'll end on is to keep building this system, what we next need to do, and I'm going to show you one way of doing this, there'll be better ways of doing this, we need to then set up that second step, right? So what I've shown you to this point is if you take an activated amino acid, intercept it with that biological acyl transfer cofactor, it will transfer the amino acid to an RNA. So we'll leave a free amine, it starts with a free amine, we're getting no peptide synthesis here. To use these structures, we then need to be able to switch on peptide synthesis. So we need in the same system to be able to control, ideally catalytically, but this is going to be stoichiometric what you see, peptide synthesis and this catalytic loading of amino acid to RNA. So you've seen this, we've come a long way from here. You've seen a chemical strategy that in water at neutral pH can take a very similar substrate, thioacid, activate that in situ to excellent peptide bond formation. And these two can be put together beautifully. So if we take a nucleic acid and a thioacid, they'll do nothing. If we add this activated amino acid, it will still transfer selectively to our diol. If then in situ we activate the thioacid, we get spontaneous formation of peptide from the aminoacyl RNA to the activated thioacid to, in one pot, in situ, under the same aqueous neutral pH conditions, make peptidyl RNA in water, directed and controlled by two simple, incredibly similar activation strategies of an acyl group. And this chemistry works irrespective, exactly the same way, whatever the base, whatever the amine, the initial donor, and whatever the side chain at the second donor to make peptide. Obviously, the way to go, what we're looking to do in the future, is understand how to turn this second step. First step can be run catalytically. This second step from a stoichiometric reaction into a catalytic reaction to be able to control coded peptide synthesis through purely chemical means. So I'll end there. Thank you guys for listening. Thank these guys for who did the work. These guys funded the work, and you guys for listening.